welcome back to the lecture series in animal physiology. So, we are into the 5th week and today we will be starting the 4th class. So, in the previous class when we discussed about the action potential, there are a few points which I wish you philosophize very clearly, because it is a very fundamental concept and there is more of a kind of an understanding what needs to be engraved in your basic framework. So, one of the thing which I wish to highlight here, so I have shown you the picture of a neuron, or I have drawn a picture of a neuron. It is a tapered one process called axon, which is the longest process, and you have multiple processes called dendrites, and you have a cell body. So, first thing you have to understand that when an electrical event happen, it could happen at different parts of the system. So, it is not that at one point the whole system is electrically active. What does that mean is, say for example, there are 5 rooms which are connected, just imagine it for your sake. So, if say, and I number the room as A, B, C, D, E. Okay. Now, suppose room A got an electrical impulse, that necessarily does not mean B, C, D, E is going to get it, but there will be some impulse will travel here and there. But if A receives a threshold signal, what we meant by threshold here is the fundamental, the word what I used, all or none. If A receives a signal which is beyond a threshold, which I was telling you around minus 40. So, if it receives a signal which changes the membrane potential of that room A by, say, take it to minus 40, then there will be a process called all or none, and which will immediately transmit a huge wave to B. So, by the time it will go to B, A will regain its own original situation, fine. A originally was sitting at a base level minus 80, right. Now, it receives an impulse and it becomes, say, minus 40, and it generates a signal which moves to room B. They are all connected. So, there is a corridor kind of thing, right. So, let me just draw it for you, that will kind of make more sense. So, today we are into, right. So, week 5 or Roman 5 or whatever, lecture 4. Okay. So, the analogy I am trying to give you is something like this. Okay. Imagine this. Okay. Okay. So, I name these rooms now. So, for example, I call it A, I call this room as B, call this room as, just touch it, you will realize it, what I am trying to tell you. C, I call this as D, okay. I call this as E, I call this as F, right. So, there are, say, 6 rooms which are attached to each other, okay. Now, I say this one receives some signal, some electrical impulse it receives, not good enough, it just spread a little bit here and there, that is it, it remained there. But say for example, this one receives a threshold, something which, say for example, this A is sitting as minus 80, there is some shift, it makes it, say, minus 70 or say minus 60 millivolt, it does not matter really, those shifts are local disturbance, tuck, that is it, nothing seriously happens. But if it receives something which could change the membrane all the way to say minus 40, then this generates a different level of wave, and that wave pattern starts to now propagate. It can propagate in both directions, it can go like this also, but depending on which direction will be favored is depending on which direction the density of the sodium channels are higher. Remember this, right? We have talked about sodium channels, potassium channels, sodium potassium ATPase pump, right. So, depending on the density in which direction will be higher, it moves in that direction. Okay, it is purely a density gradient of sodium channel which dictates the term. But then that brings us to a question, what makes nature to follow that kind of pattern is a very different question. It is evolutionarily one of the most magical thing what nature has done, how it created its chord or directionality it has given. Okay, coming back where I was. So, it generates that signal. Once it generates this signal will move to the next one, and once it moves here, to say it moves to B, what will happen by this time? The one which A at say, now let me start plotting at time t1, this was at 80 millivolt. When the impulse comes, when the impulse, that classic impulse came, this became minus 40 millivolt and may overshoot to say 10 millivolt positive. Okay. Then this will come back to its original position. By T3, it will come back to it minus 80 millivolt. But by this time, this B will exactly follow the same pattern as you see here. Then this will transmit it to C, this will transmit it to D, and overall what you will see is a wave is getting propagated. What you will be looking to is something like this with respect to time. These waves what I am drawing are with respect to time. So, there is a propagation which is taking place along these tube or along through these rooms. So, when you look at an axon, if you have seen this picture, let me put the real time axon into place. This is how. So, instead of the analogy of the room now, I will give you a technical analogy. Whenever you see these neurons, they will show you a picture like this. And let me introduce this part, which I haven't introduced purposefully, waiting for the right opportunity to introduce this concept. So, whenever you see a classic picture, you will see, just now compare this picture A here with picture B, exactly similar to room like analogy you see. So, these are the zones where you see, wherever now I am kind of these are those black spots what I am drawing are the zones which are rich in sodium channels, very rich in sodium channels. So, see for example, an electrical impulse or a signal comes here, and if it is a threshold signal, what is threshold? I have already explained you what is threshold signal. If it is a threshold signal, yes, some may back propagate like this. I do not rule out, trust my word, I do not rule that out, because there are a lot of computational models which are coming, which those of you are interested in neuroscience or computational neuroscience, they will see. So, please for God's sake do not rule that out, there is a possibility, but the major chunk, if I go by the statistics and if I say the major chunk of the signal where that will move, major chunk will move in this direction, just exactly what you saw in the analogy of the room, and they will be. So, then what are these structures? This is called the saltatory conduction, but what are these structures what I have drawn here now, which I am shading as red, room like structure. So, this is called, so a nervous system consists of broadly two basic type of cells, barring aside some cells which are of immune origin. I am not talking about them at this stage. They are, so nervous system consists of two major cells. One is the neurons, which are electrically excitable. So, I am just putting something like this. These are electrically excitable cells. The other cell type is called glial cells. Glial cells are not, do not shoot, they have certain electrical activity though, but do not shoot an action potential. So, these one shoots AP, action potential, these one no AP. And yet within the glial cells there are three classifications what we see. One is called astrocytes, we will come later about all these things, oligodendrocytes, dendrocytes, and there is a third one called astrocytes, oligodendrocytes, and Schwann cells. Schwann cells, and there is one more cell type of the nervous system which I am putting it separately, though it part of the name sounds with glia, but it is called microglia. The reason to put it separately, over the years working in these areas, kind of realize that these cells behave very differently, and if you see through the literature, if you see through their origin, these cells are mostly have an immune function, and their origin possibly is not really from the nervous, the cells which destined to become nervous system. Their origin is not like that. It seems like still I cannot tell with 100% confidence. It seems like some of the immune cells during development has kind of infiltrated into the nervous system mechanism, and they sit there because these microglia gets activated exclusively when there are neural inflammations. When there are neural inflammations like spinal cord injury, or some form of damage or some form of disease of the nervous system. Otherwise, they are quiescent or dormant cells sitting there, and their biology is not really the kind of familial properties what the neurons and the glial cells possess. They are slightly different. That is why I kind of delineate it from the main course. Okay? So, these are glial cells are astrocytes, oligodendrocytes, Schwann cells. What are their functions? They help. So, say for example, you have seen the electrical cords, right? There are so many electrical cords which are lying out here. So, one of them I just picked up. So, you see this electrical cord has a covering, this plastic or rubber covering. So, this is kind of an insulation. There are neurons which does not have, and yet most of them have it. The one which does not have will come later, once we will talk about some of the circuits which purposefully nature made it that they will have no covering. So, say for example, two axons are moving side by side like this, right. So, there is always a chance of, just like two naked cords which are rolling, there is always a chance of short circuit. So, in order to prevent the short circuit, there are insulating material, and one of the insulating material is called Schwann cells. So, that is what you see here, the Schwann cells, right. And this kind of coating what you see is a Schwann cell coating. How they are formed, that will be another day, we will be talking about this. There is a whole geometry behind formation of the Schwann cells covering. So, and yet there are parts out here which I am circling now. These parts are exposed parts, rest of it is covered, but there are, and this is called myelin sheath. Myelin sheath is formed by the Schwann cells, and these small node like structure where there is no myelin sheath presence, that is called nodes of Ranvier. Nodes of Ranvier. Okay. The way the conduction happens is, these are those excited zones. So, it is moving like this, and you really see it almost looks like the conduction is hopping. So, it moves in this direction, but then what happens once it reaches the fag end? So, now let us make the circuit a little bit more complex. Here is one neuron, see this, okay. Here is the second neuron, here we have a third neuron, likewise, okay, and the network goes on as complex as you want to make, okay. And this has its dendritic tree like this. Okay. This is the dendritic tree. Fine. Now, when a signal is traveling from here, so I said that suppose some stimulus came and it results in a threshold. So, this threshold, the action potential is traveling, the single action potential, now I am making multiple action potentials, like it reaches here. Of course, part of it will reach here too. At this zone, what is happening? Now, let us blow up this zone out here. This small part where there is, you could see a part of the dendrite, which is this one, and part of the axon, which is this one. There is a small narrow gap. I am highlighting the gap by this part. There is a gap. This small gap is called synapse, or this cleft area is called synaptic cleft. This is possibly one of the landmark discoveries of the last century, synapse, and a lot or maybe most of the truths about our very existence lies in those synapses. What are those synapses and what they do? Now, you realize one thing, unlike a cable like this, which is continuous, here we are in a situation where something like this. Now, here you can explain the transmission of information. It is continuous cable, so anything can move from one end to another end, right. But how you are going to justify this? Because there is a gap, there is a gap, there is a gap. So, this is the zone where something very interesting happen. Chemistry takes over. What chemistry does? Neurons have the other property of synthesizing a series of communicating chemicals called neurotransmitters. These neurotransmitters are synthesized in the body of the neurons, and they are ferried, like you have seen the cargo trains, they are ferried at the site of synapse. Okay. Let us draw it, that will make more sense. Let us blow up this image. Okay. So, if you blow up this image, it will be something like this. Okay. I am just blowing it up. So, this is the cell body where all the synthesis is taking place, and these neurotransmitters are put in vesicles. These vesicles, just like I told you that, like a cargo train, and there are almost rail-like structure out here of different proteins. These cargoes started to travel like this, slowly they travel, and these cargoes are filled with neurotransmitters. These dots what I am drawing are the neurotransmitters filled in the vesicles, which I am considering them as the cargoes. They are moving towards the synaptic region. Okay. So, they are kind of hopping on those rails like this, slowly moving there. And if I blow up this zone, the synaptic zone, there are two end you are seeing, right? One is the axonal end, the other one is the dendritic end. The axonal end is called presynaptic terminal and the dendritic end is called postsynaptic terminal, right? So, let's put the nomenclature in place. So, this one, which is this part. Now, let me just blow it up a little bit more. This is the axonal end. I am just putting, okay. This is your presynaptic terminal and this part, which is the dendritic end, is your post synaptic terminal. Now, at the fag end of the presynaptic terminal, the axonal terminal, what you see is, say for example, if I stretch this diagram, you will see a lot of vesicles which are sitting there like this. Okay. So, under the microscope, especially this was figured out by very good quality electron microscopy, people who were the earlier people like Sherrington, all those people, when they saw it under the microscope, what they observed was very interesting. They saw electron dense regions at presynaptic terminals. So, most of the old pictures, if you look at them, there will be something like, I have drawn it very like, something like this. If this is the presynaptic terminal, the pictures will be like this. Okay. Lot of vesicles sitting there in and around area, and there is one more organelle which is very prominent out here. So, this is how the electron micrograph images were there at that time, and this is all I am talking about 1940s, 30s, when these things were slowly getting unraveled. Okay. So, these are the electron dense zones which could be seen very close to the synaptic cleft. Now, out here there is another organelle which is very prominently observed, that is the mitochondria. There are a huge number of mitochondria which are present there. So, whenever mitochondria is present at a place, one get this feel that there must be some kind of a energy transduction, or there is a need for huge amount of energy in that vicinity. Okay. And talking about the neurotransmitters, the nervous system synthesizes a whole range of neurotransmitters, starting from glutamate to GABA, acetylcholine. Interestingly, what is slowly emerging is that during the nervous system formation, all the neurons has the potential to form all the neurotransmitters possibly, or at least a family of neurotransmitters. But as we develop, different neurons takes up or decided to stick to one kind of neurotransmitters. Say for example, what I meant by that, say for example, I say a neuron can produce say A, B, C, D and E, and A, B, C, D, E are the NTs or neurotransmitters. Neurotransmitters. Okay. Now, at birth possibly of a neuron possibly will be able to produce all these 5 in its repertoire. but as the neurons position itself at different anatomical location with different kind of functionality, they eventually will have a dominant production of say either A or B or C or D or E or whatever. So, based on that, there is another nomenclature, because if you remember the slide before, when I only talked about this classification. I did not go into the classification of the neurons, right. So, let us put the classification in place a little bit before again I get back to the synaptic zone. Based on that, neurons can be classified which neurotransmitter they are producing. Say for example, a neuron produces a neurotransmitter, say acetylcholine. Okay. So, then say A stands for acetylcholine, acetylcholine. If it is producing acetylcholine, then this neuron will be termed as cholinergic neuron. Cholinergic neuron. So, it means the major neurotransmitter which is produced by this neuron is acetylcholine. With a word of thought for you people, that does not rule out that this neuron cannot produce other neurotransmitters. Maybe it produces, we do not know, in a very trace amounts, because originally all these neurons have the potential to produce any neurotransmitters. Okay. So, say for example, a neuron is producing a neurotransmitter, say GABA, G-aminobutyric acid. So, it will be called as GABAergic neuron. Okay. Similarly, say for example, a neuron produces glutamate, then this is called glutamatergic neuron. Suppose a neuron produces glycine, then this is called glycinergic neuron. Now, likewise this list can go on and on, but there is a catch how you can classify, because that classification will help us to understand the synapse slightly better. So, I will close in here. In the next class we will talk about the classification from here, and we will talk about what happens at the synapse. Thanks a lot for your attention.